Thank you, Bill. Thank you for that wonderful introduction. It's truly an honor to be here today, and I have to say it's very humbling to be here after all of the events of yesterday. Today, I'm going to take you on a journey, and this is going to be a very special journey. It's a journey of discovery. Recently, well, let me just back up. A little bit of history. I was just recruited by Yale, so I have made a major transition in my academic life. Prior, I had been a professor of neuroscience at Columbia University, where I had been the director of the imaging research centers at Columbia. And Yale's recruitment actually created an opportunity for me to sort of tear down what I was doing and then sort of build up again something new. And one of the issues that I had become painfully aware of, actually, in neuroscience is that we were becoming very good at understanding principles of organizations of single brains. And we used to do this we do this by putting people in MRI scanners and challenging them, asking them to do things. And then what we do is look at the response from the brain during these activities, and thereby we learn about how the brain works. Well, this has been a major explosion in neuroscience in that we have learned a great deal of very useful and very important things about the brain. But the question that has been neglected in all of this is probably the question that's most important to all of us. The most important function that all of us have from the day we're born to the last day that we live is the question of what is the neural circuitry that involves connections between individuals. So what happens when we talk to each other, when we engage in a conversation? What happens together when we fall in love with each other? We have a relationship. What happens when we have an argument? What is going on in our brains? Well, we know very little about that. And one of the reasons that we know very little about that is because of our technology, that we have been enamored as scientists by studying brains in MRI magnets, where if any of you, how many of you ever had an MRI? All right, you know what I'm talking about. You go in this big tube, and it's like somebody's beating a tin can, right? And you have to lie really still. The technologist says, if you move your head, I'll shoot you. You know, something like that. You know, so you just lie there scared to death, and pictures are taken of your brain. Now, probably most of you haven't had the additional experience where you're asked to do things like look at pictures, silently name things, and so on. But you can imagine how that is. Okay, so, but you also know how impossible it is to study anything that would be associated with, related to the actual ecologically valid, as we say, circumstance where you're standing in front of somebody and you're actually talking to that person. And so my move from Columbia was an opportunity to sort of break down the old system and start something new. And so I embraced the challenge of developing a new technology. We have to have new machinery. Developing some new paradigms. That means we have to have new ways of doing these experiments. Developing some new ways of doing the mathematical computations, because we have to figure out how to stop thinking about one brain and start thinking about two brains as a unit. It's not exactly a simple mathematical formalism that we have to engage in. And then interpreting it in a way that I can get NIH to fund it for me. So we had a bit to do. And so fortunately, actually, we've made a lot of progress since that magical night that Bill and I sat on the beach with all of his board. There was the board, right? With all of his people, and my husband and I were just sort of tag-alongs. And it was a magical night, and Bill and I had a great conversation. And he sort of believed, I think, that I could do this. But I hadn't really done it yet. But anyway, we've had a year to work on it. And so I'm going to take you on a journey with me of what this year has been like, how we've been able to make progress in, I think, actually starting a new era of neuroscience that allows us to actually understand what goes on when we connect with each other. So here we go. Let's see. Okay, now, the first thing, before we go any farther, I want to acknowledge that this is done by a village of people, and I really mean this, that what I say to you today has really been the purview of the work of a lot of very talented people. In my laboratory, we say to each other often, if you know what you're doing, you should not be doing it. And we really mean it. And that means that my laboratory is sort of a bit self-selective for adventurers, for pioneers, for people who can tolerate and love uncertainty. And so you notice that a lot of my lab is pretty young. And that sort of comes with the territory, except we have a few seniors, and these guys have come with me from Columbia, and they're the best of the best, the neurocomputational guys, the engineering guys, the managing guys, the people that do a lot of the really, really hard groundwork, students that we've collected along the way, and a lot of faculty that have been extraordinarily generous to us in supporting us and helping us in various ways that only an academic would understand, but it really makes a lot of difference. And then we have always in my laboratory a number of high school students because there's something so wonderful about a laboratory that's made up of kids that don't know anything about the word no. And so we need a small dose of that, but actually it's a pretty important thing, and these guys have made pretty important contributions to us. So this is the team. What I talked to you about today has been the product of a lot of this team. Okay, so this is I'm sitting over here. I can see my slides better, and I'm kind of a wanderer anyway. So this is a picture of a very common event. We all know how to sit and talk to people. It's a very important thing. But the dialogue that we have between two people, and you can see it's a very animated discussion between these two people, sort of represents a kind of virtual connection. And we'd like to know, is this virtual connection between these two people based on biological mechanisms? If so, oops, I think I'm too far from the thing. Let's see. There, I have to stand over here. That thing doesn't transmit. Okay, if so, how does it work? And if it does work, there are a couple things we'd like to know how they work. Put it at the laptop. Thank you. Okay. All right. And if it does work, then what is the biological basis for this virtual connection? So this is the journey that we're going to go on today. Now, I want to start with this very funny, odd picture. And I hope that there's not too many of you who actually recognize this, but this is a World War II vintage Walkie Talkie. By the way, this is a sidebar. But I just learned the reason that these things were called Walkie Talkies, that they were some of the first wireless communication systems. They were quite novel at the time. But the reason they were called walkie-talkies is because there was the talkie, the guy who stood there with the device, but there was the walkie. And that was the guy, probably the sergeant, who carried the battery. So we had the walkie and the talkie, and they were a pair. Okay. All right. So anyway, but you guys didn't know. I didn't know that either. One of the techies in my lab told me that. I thought that was the neatest thing. Anyway, okay. So the reason I show you this is because there actually is a lot of similarity between this device and this device, where one is a brain and one is a wireless device. But because this topic that I'm going to tell you about today is so new, that we don't have any real models. We don't know how to go about describing this or even making hypotheses about it. We don't have any really organization. So I went back to sort of look at where are the people that have really solved these problems. And it turns out that there are engineers, and this technology developed around the time of World War II. And what's neat about this device and the brain is that there are a lot of similarities. First, they both have transmitters. One has the mouthpiece and the other has the mouth. They both have receivers. One has an earpiece, the other has an ear. And also, the human has a little bit better receiver, and the human also has an eye receiving information. So it's not unreasonable to think about these two devices as having some similar features that allow us to start a model or a way to think about this. So in addition to the features of the devices, there are also some very common features with the signals. For example, the Walkie Talkie receives and sends messages along a very low frequency range of the electromagnetic spectrum, and the ear of the human being receives signals from the audible sound pressure level, which is just a little bit higher frequency in the electromagnetic spectrum, and then, of course, the eye receives its signals from a very specific range of the electromagnetic spectrum. So they're all receiving signals along the same electromagnetic spectrum continuum. They both have transmitters and they both have receivers. And the truth is, we know a lot more about that walkie-talkie than we do about the human brain. But part of this journey is to solve that problem. So let's talk a little bit about the human system. Now, like Chuck, I have a great admiration for the peripheral sensory systems. And so what I'm going to tell you about now is the first stage of detection of wireless signals done by the human. And the human detector systems are absolutely exquisite. Let's talk about first the detector system from the human visual system. As I said earlier in the last slide, the eyes are very, very sensitive to the very specific range of signals that give us color and light and vision. But this all happens because we have a magnificent receptor system in the retina of the eye that actually absorbs this light, transduces it, and sends it onto the brain. So detector systems are so exquisite in the human brain that you can actually, under perfect conditions, detect a single photon of light. No walkie-talkie ever did that. The auditory system is just about as sensitive. We actually, again, are sensitive to a very limited range of sound pressure levels. And remember, sound comes from the fact that there's pressure of airwaves that is impinging upon the very delicate membranes of the ear, and the pressure on these membranes then is transduced to a signal that goes on from the ear into the brain for further processing. The ear is so sensitive that the displacement of a single molecule is actually detectable. So the first stage of detection in this transmission-receiver system that goes on between two brains is far better in the human being than it is in a device like a walkie-talkie, or, of course, well, I'll bring you up to speed with a cell phone. But the fact is, there are very few devices that can equal that of the human being, yet the principle of transmission and receiving is quite the same. Okay, so what goes on after the signals are detected? Well, our engineers have taught us a lot about that as well, that the signals that are detected by the holes in your walkie-talkie or now your fancy cell phone, but there's an awful lot of really neat stuff that goes on in between transmission and reception. And this is where our engineers have really, really performed for us, because they've taught us a lot about what those mechanisms are. Now, we know a very little bit about the mechanisms of the human brain. Once the signals are detected behind the holes in your head, by the way, the holes in your head is where it all starts. It comes in through the eyes, the ears, the mouth, the nose. So it's the holes in your head that start the whole business. So once the holes in your head deliver the stimuli to the brain, then the brain takes over. And it has apparatus that you can think of as a little bit like this apparatus in your cell phone. But this is, and I want to spend just a minute explaining this to you, what we know about the apparatus of the single brain that processes language symbols. And first, it's an opportunity to sort of make the point that the first principle of brain organization is the principle of functional specificity. And that's a very fancy word, but it simply means specific things in your brain, specific parts of your brain do specific things. And so the brain's pretty smart. It divides up its tasks, and it has little departments that are specialized for receiving sound information and making sense out of it, for transmitting sound information and giving you the ability to talk to somebody else. It has some processors that allow you to kind of manipulate the material in between and so on. So here, at the back part of the brain, which is sort of the receiver end of the brain, we have the visual system and the word comprehension system. We often call this Wernicke's area because it was discovered about 1850 by a physician who discovered that patients with lesions in this area could speak perfectly well, but they didn't make any sense. And so he termed this an area for word comprehension. The vision area is a very specialized area, and I want to make the point here that the visual system is that's relevant to us and this meeting, and also I'm going to tell you that the visual system is also divided into specializations. And one of the really important specializations is specializations for faces. And I was so impressed yesterday when David White was talking about the sensations and experiences he was having looking at the face in the stone. And what I was thinking about was what that said that was so beautiful about the human brain, because the human brain is so sensitive to faces. Faces are like the most important visual system that you see. And in fact, we know that because the brain has devoted a tremendous amount of territory just to processing faces. So a happy face, a sad face, a fleeting face, an old face, a young face, a disturbed face. Faces, just anything that looks like a face, a cloud that looks like a face, the moon that looks like a face, anything that we can make a face out of, we will, and we give it meaning. And that's the purview of the brain. That's what the brain does with that information. So it's a very beautiful receiver system, receiving both auditory and visual information back here. The transmitter systems up here having to do with what we call Broca's area, again, relating to the discovery of the area based on lesion studies. And so in this brain, we can think about receivers and transmitters and stuff in between. And that's what we're going to talk about, the stuff in between. Now, okay, the stuff in between. So we know a lot about the stuff in between, as I said earlier, because we know how to run functional magnetic resonance imaging machines. And this is an example of the way we know about the language system. So we put somebody in the scanner, and by the way, this was my scanner at Columbia, and one of my students, and look at the holes in his shoes. I mean, really? Really? Well, I never undid that picture. One could Photoshop it out, but, you know, it's sort of cute. Anyway, it's just real life, you know. Anyway, so we asked these guys in the scanner to name pictures. And so they're in there, they're looking out through a mirror, and we show them pictures. And since they can't talk in the scanner, we say silently to yourself, I want you to name these pictures. I just sequentially just present pictures. They name them, and when they do that, out comes this beautiful picture of the brain. It's not quite that beautiful when it first comes out, but it's sort of like that, where we get all of the natural parts that process language. But this, of course, doesn't tell us anything about what goes on when we actually talk to each other. This is just the language system when it's perfectly static. And so functional imaging allows us static views of single brains. So now let's see what, okay. There we go. Okay, so now, enter the stage with a new technology. And this is what it's so much fun to tell you about today. First of all, this is Adam. And Adam is one of my engineers that came with me to the lab. This is the cap that we call near-infrared spectroscopy. And this is the substitute for the MRI scanner. And near-infrared spectroscopy actually measures brain signal by injecting little lights into the brain and then measuring the reflectance of those lights so that we have a message of about how much oxygen is being used by the neurotissue that's underneath each of these little flashlights. I'm going to say a lot more about that in just a minute. But just moving forward with where we've been, so we have our people put on these little helmets, and then they do the same object naming task, and we can measure exactly the same things, but we do it in multiple people, and we can have them doing it together. And so this is the major technology breakthrough. Now, near-infrared spectroscopy has been around for a long time. I didn't invent it. I resurrected it. It's been around. It was discovered by Britton Chance around 1981. And it never really took off because, again, functional MRI was so successful. But it has been used clinically, and sometimes it's been used because it's so safe. It's been used to study brain development in infants, but it's never been used to study brain activity in cognitive events. And one of the reasons was that there wasn't the ability to image the whole brain. You notice this is a cap that has what we call optodes, not electrodes, optodes, all around the brain. And so one of the things that I did was that I went to Japan, and a company by the name of Shimatsu, maybe you know them, agreed to make me a special machine that would would do a whole head set of optodes on 2 brains. And so Shimatsu deserves an awful lot of credit. These people thought I was nuts. They thought I was just nuts. But for some reason, they agreed to do this for me. And so now we have the world's largest 2-brain near-infrared spectroscopy system. And so this is a little bit how it works. And so I'm going to tell you how we measure the language system of 2 brains working. And then the road is going to get a little bumpy because I'm not going to spare you guys any details. So I'm going to tell you really how it works. And I'm going to tell you really what the data look like. And I'm going to tell you really how we analyze it. And you're going to hear a couple words like wavelets and kernels and stuff like that. But it's okay, because I'll take you out at the other end, and you'll be able to see kind of what it all adds up to. So if you don't get it all, it's okay. We'll get you there conceptually. Okay. So this is Jenny in my lab. She's a Yale undergraduate, and this is Adam again. Jenny's talking to Adam, and Adam is ignoring her, which, it's not the first time it's happened, probably. Okay. So this is what they're doing, and this is actually quite important. We're going to measure now 2 brains simultaneously doing a very simple language task. And we're going to do it in 2 ways. We're going to do it in a way that we call monologue, and that is a way in which they're just talking and listening, but there's no interaction. And then we do it in a way that they talk and listen, but they talk and listen to each other so that there is an interaction. And what I want to see is the neural circuitry that differs between the monologue and the dialogue, and that's going to give me some information about what goes on when 2 people connect. So this is the monologue condition, and we're using the old Boston, what we call the Boston naming or object naming task, because we have known for 20 years that this is the task that actually identifies the language system. So here we go. So subject 1, which is Jenny here. She gets her picture. Pictures come up on the monitor. She gets her picture. It's a clock. Jenny names it as a clock. She talks about the clock. Adam supposedly, hopefully, listens. And then Adam gets his picture, and it's a coconut, and he talks about his coconut for 15 seconds, and Jenny listens. And then 15 seconds comes up, and she gets her picture. It's a camera. She names the camera. She talks about it, and Adam listens. So 15-second epics, talking and listening, until they get done with the whole experiment, which is 3 minutes and 12 seconds. Okay, perfect. So we collect all this data. While we're doing this, every single optode on their heads is sending data to a central computer. Now, we have them do the experiment again, and this is in the dialogue mode. So we start out with Jenny talks about her clock. And then Adam, when his picture comes on, he doesn't talk immediately about the coconut. The rule is that he now talks to Jenny about the clock. So he's responding to her about the clock, and about halfway through the 15-second epic, then he is allowed to talk about the coconut. And when he talks about the coconut, then Jenny just listens. But then she gets her picture, but she doesn't talk about her picture until after she responds to Adam about the coconut. So they go through this whole thing with talking and listening, but responding to each other about their pictures. So there's not a lot of interaction, but there's a little bit. And that difference that we're going to see between the brain signals representing a little bit of difference is going to be really important to us because it raises the confidence level that we've got it right. All right. So basically, we've done this experiment with We've actually done 2 experiments. So the first one I just described you, the verbal transmission experiment, we've done it in monologue and dialogue. Now we're going to turn around and do the exact same experiment again, but this time we're going to allow people, as they were, when they were doing this, they're doing it face to face. We're going to do it again, but we put an occlusion between them so they can't see each other. And this experiment allows us to understand a little bit about the neural events that are associated with the nonverbal cues of language. You know perfectly well that when we're talking to each other, we don't just give each other spoken narratives, that we wave our hands and we smile and we sort of have this body language, that, whatever it is, that says things that's above and beyond our messages. And so we really want to understand the input and value and the impact of the nonverbal communications as well. So doing this face-to-face gives us sort of a hint about that. So we've got the talking experiment and we've got the face experiment. Okay, now, what about the signal? And this is when I'm not going to let you guys off the hook. You're going to have to hear what it is that we are measuring, because this is really cool. This is not magic. We don't just put little caps on the head and just know exactly about everything what the brain is doing. The brain isn't so easy to figure out. We have signals, and these signals are a little bit hard to decode, but we collect them in a certain way, and then we figure out what they're telling us. So the signal from this type of work, and it's the same in fMRI, comes from a first principle of physiology. And that first principle of physiology is that with any working neurotissue, that is, any part of your brain that's working, that it is recruiting blood to that area, and specifically to that area. So as neurotissue works, it requires metabolic support. So it recruits blood to that area. It's just exactly what you would expect. If you didn't know anything about it, that's what you would say. Well, that's what it does. But there are two things that happen with that. The first one is that in the local area that's working, there's an increase in oxyhemoglobin. Now, oxyhemoglobin is the blood that carries a lot of oxygen. And why does it have oxygen? Well, it's coming right from the heart and the lung. It's got a whole lot of oxygen, and it's delivering it to those working neurons that need the metabolic support. Okay, the second thing that happens is that in that local pool, there's a decrease in deoxyhemoglobin because as you're pooling in all of that oxygenated blood, proportionally, the blood that doesn't have the oxygen in it is reduced. So there are two things that happened because of this first principle. That gives us two signals. Now, all right, so imagine this situation where you have a brief event. Let's say a flash of light, and the light comes on for 2 seconds. It's just bam, on, off. Then you're reading from an optode that's looking at what's happening at that moment. The first thing that happens is that there's a whole lot of oxyhemoglobin that comes in. There's a whole lot less of the deoxyhemoglobin in that local area. And those are the only two things that are happening. We like to measure the total, which is just the sum of this and this. And I put this here just for fun because if you were in an MRI machine, you would be measuring this deoxyhemoglobin signal only, but it would look like that because in an MRI machine, we invert everything. It doesn't matter why. It's just that it's the same signal, it's just upside down. That's all you need to know. So there's only two things. There's only two things that really happen there. Okay, so we've got an increase of oxy and a decrease of deoxy, which is pretty cool because we can use both signals. Now, how do we get the signal in the first place? Okay, well, I'm going to get the whole picture on here. Basically, these optodes, these things, these little flashlights that we put on the heads, have little lasers in them. They're not just flashlights, they're lasers. And these little lasers have very specific frequencies. And so we inject these little frequencies into the brain. And in fact, we actually inject 3 frequencies, one that is sensitive to the deoxyhemoglobin, one that is sensitive to the oxyhemoglobin, and one that is sensitive to both equally. So we're injecting wavelengths of light, and what we're doing then is measuring the amount of absorption for each of those blood types. So here's kind of how it works. We've got a brain, and then we populate it with flashlights. And those are optodes. Those are what we call emitters. It's a fancy word, the little flashlights with lasers on them. And then we populate it with detectors. That is, we have to have little units that tell us how much of those wavelengths we're getting back, because we think that the signal of how much oxygen and deoxygen, how much oxy and deoxy is changed is valuable to us because it's telling us about what's going on in the brain. And so then we have what we call a bunch of channels that are in between, and any combination of those emitters and detectors gives us a channel. So basically, the story is just what I said. We've got emitters, 3 different wavelengths. We've got channels in between, and detectors. Now, for any of you out there, most of you don't want to know this, but the detector doesn't actually, so nothing's easy in this business. The detector doesn't actually give us exactly what we want to know because it's just a measuring absorbance. So we have to do some fancy calculating to give us a signal that tells us about the concentration of oxy and deoxyhemoglobin. So okay, so we do that. And then a couple other limits, the things I want to tell you about here, and then we'll go on. We'll just assume that you guys know how we get the signal and what it is. But we're acquiring those signals at 33 milliseconds, which means we're acquiring them very, very, very rapidly. 33 milliseconds is on the order of magnitude of EEG. EEG is a little bit faster, but it's really, really good as opposed to about the 1.5 seconds for functional imaging. But the spatial resolution sort of sucks. Spatial resolution in an MRI is about the size of a grain of rice. So we tile the whole brain with very, very small little things like we call 3-dimensional pixels, like in the camera, your pixels. These are voxels. But here, our pixels are about 3 centimeters. But it's good enough. And so So it gives us a good picture of what's going on in the brain. And I'll tell you, the advantage of being able to image two people simultaneously as opposed to that very fine resolution seems to me like a pretty fair trade-off. So that's the trade-off that we've made here. Okay, now I'm going to take you one more bump in this journey, and then we'll start our way home. It'll be easier. But I'm not going to let you guys get by with not seeing this signal. This signal is just knock-dead gorgeous. You have to look at it. You have to see it. That's all there is to it. So what I'm going to show you is the real nuts and bolts here. And I'm going to take a raw signal. This is Diana from my laboratory. And she's wearing the full Shimatsu head cap. And she's just doing this very simple talking and listening task. And again, this is still only single brain. The best is yet to come. But I'm just telling you that what we do, of course, with single brains, we, of course, do with two brains together. But you need to kind of understand this. So Diana's doing this task. She's talking and listening, and so she talks for 15 seconds, she listens for 15 seconds, she talks for 15 seconds, listens, blah, 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 all the way through it. Okay, and so then what we're doing is reading the signal from one channel right there. Okay? Bam, there it is. So, gorgeous. It's a beautiful thing. But it sort of sits there and it's kind of dumb. We have to, this is the way signals are. They don't really tell you what's in them. You have to kind of push them around a little bit to sort of get the message. But anyway, let me just document for you that we're getting what we think we should have. We're measuring two signals, the oxy and the deoxyhemoglobin content during the talking and listening. So Diana's talking for the first 15 seconds. The oxy goes up, she listens, and the oxy goes down. She talks again, the oxy goes up, she listens, it goes down. Up and down, talking and listening. The deoxyhemoglobin, as you saw on the slide prior, is exactly counter-correlated. It goes down when the oxy goes up, so the signal goes down when she's talking, up when she's listening, down when she's talking, up when she's listening, blah, 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 the same thing. Gorgeous signal, and it happens with every single optode across her entire head. Now, one thing I want you to notice about this signal, if I can get it back here, is that there's a lot of squigglies in this signal. And it's in these squigglies that the information is. There's no information in the fact that it goes down and up because I'm driving it, right? There's no information there. What the information about what's going on in the brains is going to come from these little squigglies. And so the next part of what I tell you is how we decode this signal to get information about two brains. Okay, but I think I've got, one more slide that's kind of a sidebar here. And I probably didn't need this slide for this group, but if this was a real science symposium, I would need this slide because I would need to document to you that when we're doing this two brains at a time, and now I'm going to talk about a group of people, and we ran 34 people, which is a pretty substantial group of people for an experiment like this. That put their brains side by side. By the way, for those of you who aren't used to looking at brains, this is the front of the brain, the top of the brain, bottom of the brain, back of the brain. And so I've got two brains looking at each other, so they're a mirror image to each other. And these yellow numbers are the locations of the channels with our optodes. And here I've just highlighted corresponding, or what we say, homologous channels across brains. And these channels all have names because we have a standard atlas. And so I can digitize each of the locations of those optodes so I know exactly where they are in terms of the anatomy of the brain. In fact, I can even give it an address in terms of formalism that allows me to put these brains on a Cartesian coordinate system. And so then I lump all of the brains together, and I look at all the brains for every single channel. And I'm only showing 4 of them here, but this is for all. In this case, I'm looking at 30 channels. That when all of subject 1s are talking and subject 2s are listening, you see the form is here. the talking goes up, the listening goes down, and they switch at 15 seconds, and then talking is up and listening is down, and so on. And if I show the other signal, deoxyhemoglobin, they're in exactly the opposite format. So this is just reassurance that we've done what we think we've done, and that the signals are as good as they can be. All right, so now this is the nuts and bolts. What in the world do we do with that signal in order to understand something about the neural circuitry between two brains? That's why we're on this journey, right? We want to know what goes on when two brains talk to each other. And does that mechanism represent something about the coherence, longing for coherence between two brains? So this is the last part of the journey. So what do we do with the signal? I want you to imagine that we're going to talk mostly about this signal. It doesn't make any difference, but the example I'm going to show you is the deoxy signal, because the deoxy signal is for reasons that I don't have time to go into, is the signal that's arguably the most closely related to the metabolic activity of the neurons that we actually want to study. Okay. So I want you to imagine this signal with all of its squiggly lines is made of porcelain. And you can hold this signal in your hand, and then you drop it. And it smatters everywhere. You've got this porcelain shards of signal all over the floor. And then somebody comes along and gives you several little magnets. And each of these little magnets are of a particular size. And you take the magnet that's a small magnet and you just run it along and you pick up all the small pieces. And then someone gives you a little bit bigger magnet and you go along and you pick up all the little bigger pieces. And so on until you have an array of magnets and you have all of your signal picked up, and each of your magnets contains little bits of that signal. That's wavelet analysis 101. And if you guys got that, you just passed the first class of engineering wavelet analysis. Okay. And actually, I don't think that my professors that taught me that would have liked that analogy very much. But I bet you guys get it. Okay. So now, this is what happens. So this signal right here, that's the one I just broke to bits. And these are my little magnets. We call them kernels. For any of you guys who know anything about physics and mathematics, these are kernels. You know, they're not magnets, they're kernels. But anyway, think of them as magnets. And then this are the little bits and pieces that I pick up off the floor for each of those sizes. And if I put all of this together, if I glued it all back together again, I would get this. So there you are. I just smashed the signal, and then I could put it back together again. So that's how we begin to figure out what's in that signal, because in that signal contains all the information that we want to know about what's going on in the brain. We just have to be smart enough to figure out how to decode it. And so that's the first step in decoding it. We smash it all to bits, we look at all of its components, and then the magic happens. Okay. So, okay, that's what I just told you. We smash it all to pieces. And then, okay, so this is what we do with all the pieces. That we have each of these channels, and each channel has a signal, right? So each channel has one of these porcelain signals that we break into bits and pieces, and we pick it up. So every channel has an array of these bits and pieces. And then every other channel in the brain has the same thing. So what we do is we ask for any given channel in one brain, how do those signals cohere? How are they in synchrony? How are they entwined with? How are they correlated with the signals in the other brain? Now, are any of you guys in IT out there? Some of you guys, we got some IT people. Okay, I just want you to now imagine how long it takes 96 processors, half a terabyte of RAM, to do this computation for 34 subjects? The answer is about 2 weeks. 2 weeks. And so we just let this thing rip and go do something else. And eventually an answer comes out. But the goal here is to compare the extent to which wavelets, those are the little magnets, wavelets from one brain cohere or covary with the wavelets from another brain for each of the conditions of our experiment. So remember, we want to differ. We want to look at how the monologue and the dialogue and the face-to-face and the face-occluded differ. And what is really cool about this, and this is what's the fundamental difference between this technique and EEG, is that I know the source of every one of those signals, because I can ask this question now with respect to the very specific neurocircuitry of the brain. So, okay. It's easy from here. You guys can take a deep breath. That's the last of the hard slides, and now it's going to be fun and easy. All right. So, just go back and remember that we've got two people, and we want to understand what goes on between the two of them when there's a dialogue. So there's only two functions of interest to us: transmission and reception, and transmission and reception. Okay, that's it. It's that simple. But as a good scientist, we have hypotheses about what we think the data should look like. And it's a good idea to have a hypothesis, but you kind of got to take a hypothesis with a grain of salt a little bit, because the data really rule, and sometimes your hypothesis is wrong. But nonetheless, in science, we kind of like to know a little bit about why we're doing this and what we expect. And of course, there's always a rich literature that's gone ahead of us that gives us some insight. And the insight, I already told you that what we know about single brains, it sort of leads us to this hypothesis that we would expect that the known receivers in our brains, that is Wernicke's area or the face area of the brain, would covary across interacting brains with the transmitters. So what we would expect is that when I'm talking to you, that information is resonating with your parts of the brain that are receiving that information, and when you're talking to me, it goes the other way around. So that's the first expectation that we would have. Now, what does that mean? Very simply, what we think that means is if this hypothesis is confirmed, that we would actually take this as this covariance between our two brains as evidence for neural processing that tells us about the interpersonal interaction. And this would be the first time that the neural processes that were joining two brains as a single unit would have been discovered. Okay, so those are the ground rules of our experiment. Results. You ready for this? It's pretty cool. Okay, so we waited 2 weeks now, and the computer has finally spit out its results. It's a little bit like, did you guys see the movie about Enigma? Okay, and they waited and waited and waited for Enigma to give them results, and finally they had to get a little bit smarter before they figured out how to make it work. But anyway, it came out with the results. So finally, computer gives us these results. And here's a picture of the results, and then, of course, I'm not going to spare you the raw data. You're going to have to look at the raw data with me. But if this were going into a textbook, this is what it would look like. The hypothesis was very, very nicely confirmed that these two brains, and this is consistent, of course, with everybody in the group. This is a group study now. Between their Wernicke's area and Broca's area, and vice versa, these two areas of the brain responsible for transmission and reception are working together like pistons in an engine. And that's what the data analysis with the wavelets told us. And I'm going to tell you how it told us that. So let's go back and look at the single brains. Oftentimes there's such insight that comes from looking at data from single brains. We like groups, we can generalize from groups, but from single brains, we can really understand what's going on. And so we have a common format here for displaying data. It's called a heat map, and it puts things in colors so that it's a little bit more understandable. So this is a single subject, and this is this particular combination of areas. So it's one person's Broca's area and the other person's Wernicke's area. Now remember, all other parts of the brain were actually sampled. This is the only result that came out. Every possible combination. Imagine the matrix. Brain areas from subject 1, brain areas from subject 2, and all of the areas in between were sampled across the entire range of wavelengths. The only result for the monologue-dialogue experiment that was statistically significant was the one we hypothesized, Wernicke to Broca, Broca to Wernicke. And this is an example of that from just one sample subject. So what's plotted on this axis is the wavelet length, is the specific wavelet. Remember, we broke up the signal into all of these bits and pieces, and each little magnet that we picked it up was a specific wavelet. And so, first of all, I want you to notice that the color on the monologue line here across the whole time course of the experiment is less than the color on the dialogue line. I mean, what that tells you is that the two brains are a lot more correlated in dialogue across the entire experiment than they are in monologue. Now, the additional piece of evidence here is from the whole group. And that data, those data are here, which is really kind of cool. This is the wavelet now, and this is the coherence. And this is the axis that we're really interested in. This is how much the two brains are in synchrony with each other. And you'll notice, and this is for each area of the brain, that the dialogue line usually wins. And it's particularly significantly different from the monologue at a wavelet of 6.32. Now that's really significant because that's halfway in between our epoch of 15 seconds. Remember, people talked and listened and talked and listened, and that was 15-second epochs. And halfway in between that, they switched from my picture to your picture, my picture to your picture. And so that switching would be expected to be where the coherence between the two conditions would be the highest, and it would be about midway in between the wavelengths. So this analysis here is really most convincing, that this result is really the first result of coherence between two brains. Now, we did a second experiment as well. Remember the vision experiment. And the bottom line is really similar in that we have actually two results, but we've got, of course, fusiform, which is the face area of the brain, co-varying with the partner's dorsolateral prefrontal cortex. Now, I haven't said anything about dorsolateral prefrontal cortex. It's a very popular area in the brain. It's in the frontal lobe. And the dorsolateral prefrontal cortex sort of does all possible things, which is why people don't talk about it very much, because it does everything. So everything you talk about involves the dorsolateral prefrontal cortex. But the fact that it does all possible things makes it kind of interesting, because it's not a transmitter. It's a processor. And so remember, my hypothesis was that we had to see covariance between areas that were connecting reception and transmission in the brain. Well, it turns out that there is another result here. So in this case, there are two results. And the second result was simply that the dorsolateral prefrontal cortex then connects to Broca's area, and these two systems go at different time scales. So we go from reception to transmission via the dorsolateral prefrontal cortex. The eye stuff, the face stuff, goes very, very fast. You know how when you're looking at people, you get that information so fast. When you're talking takes time. Words are long, and information and sentences, it takes a long time to process. But if you're just looking at somebody, you're getting information just bam, 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 really fast. And so that's what this says. We've got two processes, the face, the fusiform face area going to this processing area, and then the area that's associated with Broca's area, which is the speaking area. So this process is a little bit more complicated. We get fusiform connected to the transmission area, that the synchronous exchange has 2 time courses, and that facial information is embedded very specifically into this language system as we understand it when we're talking back and forth to each other. Okay, so what does this mean? Summary. Isn't that a great word? Summary. We're getting there. This journey is almost over, and it wasn't so bad, I hope. But okay, so the summary. Two brains as a unit, and that's really kind of a beautiful thing because that's not something that's ever been done in neuroscience before. And so the finding here, I think, is that these are wireless interpersonal communications, like your walkie talkie, like your cell phone, go on between 2 brains, and that this actually integrates the functions that are going on into 2 brains under the grand task of transmission and reception. So we have receivers, Wernicke's area and fusiform areas, just like the walkie-talkie. We've got transmitters in the brain, and we've got processors, things that do all possible things in between the 2. And there's probably a lot more, when we get a lot more sophisticated with our experiments and this line of work goes on, we'll learn a lot more about this area. This is a very crude, rough sketch, but this is what the data are right now. So what we've seen is that between the 2 brains, that those receivers are connected, the transmitters are connected, both for the auditory system and for the visual system, and we can begin to think of 2 brains as a single unit. 2 brains as a single unit. I mean, this is really, really amazing, that 2 brains become one, and that is a very, very, I think, profound model for all of us in neuroscience. But what I learned yesterday from other people's talks, that this might have some profound impact also as we begin to think about connections between all of us and an evolution of consciousness and so on. And I'm actually going to use the word consciousness in about a minute. So stand by for that. Okay. So I want to go back to my first slide, and where we asked the question, how does it work? What's going on between 2 individuals when they're having a intense conversation? First, as you know, there's a transmission and a reception stage, and then it goes the other way around, transmission and reception, so it is reciprocal. It's going back and forth. So we have this reciprocal wireless. It's on the electromagnetic spectrum signals that communicate data across brains via the visual and auditory detection systems. And again, reminiscent of Chuck's statement yesterday that the sensory stimulation was like deep brain stimulation. Isn't that what you said? Deep brain stimulation. Okay. Well, we don't get too deep, but we're stimulating the brain with these transmission and reception signals. These signals are well known. The detector systems are probably the most understood parts of the brain, and they're working as I said before, like pistons in a machine and an engine back and forth. Okay, so that's the first part. The second part is that I use the word covariance, but I mean synchrony, the extent to which the signals across brains are the same, that the covariance of these reciprocal reception and transmission processes is evidence of the connection between 2 brains. It's really important to understand what the evidence is, what I'm calling is evidence for connection between 2 brains. And so we've got these 2 people that are transmitting and receiving, and that their brains The brain areas specialized for reception and transmission are in a coherent or synchronous mode during those moments of conversation or intense interpersonal interaction. So what's the new idea here? Well, there's lots of new ideas. But I like to think of this aura, this connection between two people, as something like an effective social synaptic junction. A social synaptic junction. Yeah, laughing. That's a good response to this. I think that's what my husband would do, but he's not here. But I love it. A social synaptic junction, because it describes a process. It gives a process a name that we've just discovered that I think might help organize sort of our future thinking. So just hang on to that idea, social synapse between two brains. It's just an idea to pin all this on. So my original question, is there a neurobiology of collective consciousness? Well, I don't know the answer for sure, but I do know that we have shown that there's a biological infrastructure, that there's a biological mechanism for this reciprocal transmission and reception that connects people together during a conversation. That we know because of what I've just showed you. So what does that mean? Well, maybe. See, I told you I'd use the word consciousness. Maybe, that this is the biological we that some of you spoke about yesterday. Maybe this is the biological we that could be the basis for an emerging or collective consciousness. So I just leave you with that idea. Thank you. I did it on time. Question, yes.
Could you talk a little bit more about the relationship between what in one slide you called entrainment and otherwise called coherence?
Yes, okay.
Did everybody hear
Yeah,
that?
I asked about the relationship between entrainment and coherence.
Right. I was a little bit confusing. I've used a couple of words and I mean the same thing. Coherence is a mathematical term that really describes how two signals vary together. And to the extent that they vary together, the coherence is high. And if they don't vary together, if they're randomly related, the coherence is low. I use the word entrainment and also synchrony to describe the same thing. What I'm describing is the way in which signals from the brain do the same thing. And mostly they don't. But in the cases of interpersonal dialogue or face-to-face, that our brains are entrained. Yeah.
So just to make my question clear,
Yeah.
I've done some reading about social entrainment,
Yes.
which happens, for instance, when everybody sings together.
Yes.
So how is that related to what you've been doing?
Yes, okay, great question. Of course, we've done, we now are exploring other questions. I don't have final results, but we have now a line of work going between musicians that are playing instruments back and forth to each other and actually doing that in synchrony. We plan to have people singing together. We don't have that yet. We have people in arguments and in neutral experiments. We have people competing with each other, playing poker with each other. I mean, my guys. Anyway, it's got to be poker, right? And these guys are like killing each other. You can't believe what's going on right now. Anyway, I just hope that the IRB doesn't come and complain at some point. But anyway, so we're looking at what happens, how does this change under different conditions of interaction. And singing, synchrony, is one of those things that's really, really interesting. Just a sidebar here. The preliminary findings with music as people are playing, in this case, it's keyboards. They're playing keyboards in monologue, just your snippet, your snippet, your snippet, your snippet, and then they're playing and improvising, playing to each other in response to each other. The brain areas that are responsive and cohering in those conditions seem to be spread all over the brain. It's, this is so preliminary. I haven't hardly written it down yet, so don't quote me on this. But it's really interesting that when we compare the spoken word, the language, which is a system that's very specialized in the brain, with a similar paradigm that has to do with music, it seems to be very distributed throughout the brain, which is a very interesting comparison that may take us a long ways in understanding value of music. It's a very good question. Thank you.
So you mentioned angry at each other. And did you have a sincere anger experiment where people really are in the fight with each other?
No.
And the other side of that, have you had people with the apparatus on when they were making love with each other?
No, we actually haven't done that yet. No, we haven't done that. Shoot. But, and in the case, we don't, in our current experiments, we don't have people that are really angry with each other. We have people disagreeing and agreeing, and we're comparing that. And we are seeing difference when we'd like to get emotional components. Actually, I had a lovely discussion with David White last night, and he said, what would happen if you had two people that were reading poetry to each other? Wouldn't that be a nice sort of understanding? Because the poetry is so emotional and so deep that it would be an opportunity to actually tweak this a little bit, to look at the brain. Anyway, thank you, a great audience. I just want to tell you.
I have one question.
Oh,
just
okay.
the correlation, and you were talking about it now, the correlation between the heart and the electromagnetic field in the heart and what is happening and where is our brain?
Okay, this is a very, very important question. You know those little blips? I said there's information in those blips. You can read the heart rate perfectly from those blips, and we do it. It's perfect. That the heart is in the brain every single beat.
Thank you.
Great.